Hey guys, welcome to the Chapter 12 lecture video. So Chapter 12 is the first chapter about the immune system. So our immune system is really large and complex. You'll notice the subtitle is Host Defenses Part 1. So there's actually going to be a couple of chapters to get through all of the immune system. I'm going to move my face around during this recording so it's not in the way of the lecture. Okay, so there's 3 lines of defense of the immune system. The first line, second line, and third line. So the first line of defense is any physical barrier that prevents microbes from getting in. So skin is a classic physical barrier. It physically blocks microbes from getting into our internal organs. The second line of defense is also known as the innate immune system. And the first and second line of defense is what we're going to cover in this chapter. And then in the next chapter, we'll talk in depth about the third line of defense, which is also known as the adaptive or acquired immune system. And this is a learned response that we build in response to specific pathogens, and we create memory in the third line of defense. So this is just another slide showing you in sort of a visual form. This is just an image from the textbook that I really like, showing you the first line, second line, and third line of defense. So the first and second lines are considered part of the innate or non-specific immune system. These just recognize they just generally block foreign things. They can't specifically identify things as like flu or E. coli. They're just like foreign and they block it. The first line of defense that we'll talk about in this chapter includes physical barriers like the skin and chemical barriers, any chemical secretions that we produce that kill microbes that are trying to get in. And then the second line of defense, which is a bunch of white blood cells whose purpose is to basically cause generalized inflammation. All right, so we'll talk about the different chemicals they produce that generate inflammation and the steps of inflammation. Fever is kind of a part of inflammation. And then the other big thing that's part of the second line of defense is phagocytosis, or eating of foreign particles by certain white blood cells. And then next chapter, we'll talk more about this third line of defense, which involves a whole nother set of white blood cells, primarily our lymphocytes, our B cells and T cells. But that works hand in hand with this second line of defense. So it's kind of a chain reaction that we'll see. So some of these physical and chemical barriers, the first line of defense, includes the skin. So the outer layer of your skin is just a bunch of dead skin cells, basically. So they can't be infected because they're dead. And they slough off, so any bacteria or virus particles that stick to them just come off when they slough off. So exfoliation, natural exfoliation, is sort of a natural shedding of your old skin, but also of any microbes that are stuck to it. You also have sweat glands and sebaceous glands that secrete chemicals onto your skin, and those chemicals can be antimicrobial. So sweat contains an enzyme called lysozyme, which is also found in tears, which are produced by your eyes. The fluid from your eyes contains this enzyme lysozyme, which actually damages some bacterial cell walls. So it's a type of chemical barrier. The digestive system is open to the outside world. You take food from your environment, you put it in your mouth, it goes into your body. So that's a pretty vulnerable sort of internal body structure because it's open to the outer world. And so you need to have a lot of defenses there. So you've got saliva with enzymes in your saliva that help to digest food, but also can digest microbes. You've got the constant movement and flow and flushing action of the digestive system. There's tons of enzymes and acid in the stomach that their main job is actually to help sterilize your food before it moves into the intestines. And then that flushing action can also work in reverse. If you have an infection, vomiting is a way to expel pathogens. The respiratory system is also open to the outer world. You're breathing in microbes every time you take in air. And so you have to have a system to trap them or to expel them. So in the nose, in the nasal passage, you have hair, mucus, you've got these turbinates, which are like these little pieces of cartilage that the air spins around so that it has the opportunity to get stuck in those hairs and mucus. So it really helps to trap particulates in the air you breathe so it doesn't get down into the lungs. And then even in the airways in the lower respiratory tract, like the trachea and the bronchi, you've got these cilia. The cells there are ciliated and they produce mucus. And I'm like doing this thing with my fingers. This is called the mucociliary elevator. Basically, the cilia are constantly pushing this mucus up and out. When you have a cold, you produce a lot of mucus, and so you're constantly like you can feel it. You're coughing it up. And maybe spitting it out. But when you're healthy, you're still doing that, just to a lesser extent. It's always moving up and you're always swallowing it. Every time you swallow, you're swallowing some saliva, but also some respiratory mucus that's been coming up from that mucociliary elevator. And then you swallow it into the stomach where it gets all, digested in the gastric juice. And then lastly, the genitourinary system, another body system that's open to the environment and outside world. So you do have to have mechanisms of keeping it sterile or keeping it free of pathogens. It's not always sterile. So the urine, first of all, you pee multiple times a day. That constant flow has a flushing action and helps to physically flush things out. The pH of the urine can be inhibitory to some microbes. Seminal fluid actually contains several antimicrobial compounds and also pH buffers. So both of those things can contribute to the lack of growth or prohibiting the growth of bacteria. And then the vagina actually has a naturally acidic pH, and that is to promote the growth of appropriate bacteria and to prevent the growth of pathogenic bacteria, though changes in vaginal pH can change the vaginal flora and lead to infections or vaginitis. Some other physical barriers besides the skin and physical tissues include our actual microbes, our gut microbes, our resident microbes, so skin microbes as well. Remember, I showed this picture in a previous lecture that our normal flora sort of act as our territorial protectors. And so they are really important for not only physically blocking pathogens from colonizing, but also they produce some antibiotics. And as you saw in that video from chapter 11, they're really important in training our immune system, particularly those B cells and T cells of the adaptive immune system that we'll see next chapter. This picture here is, there's sort of this theory of dysbiosis in the microbiome world, where the concept is that when we have a rich and diverse set of normal flora, normal bacterial, then they train our immune system properly. We have a healthy functioning immune system, and we've got a rich population for bacterial antagonism. All right, but when we deplete those species, and especially key species, which we're not sure what those key species are yet, then it leads to dysbiosis, so an abnormal collection of microbes, and that can lead to improper training of the immune system, which can lead to immune disorders, things like allergy, autoimmune disease, and potentially increased risk of infection because of less microbial antagonism. So this is sort of a theory that's being tested, but this concept that our microbes are important for our health, and when we don't have the right populations of microbes, we have disease states. So that's the first line of defense. The second and third lines of defense are much more intense. So immunology is really the study of all of the second and third lines of defense, the innate immune system and the adaptive immune system. Also, part of the immune system or immunology is the study of allergies and cancer. Cancer is actually something that the immune system patrols and kills cancer cells. So the development of cancer as a disease pathology is really a failure of the immune system to recognize and destroy those cells. So we'll get into that a little bit more in chapter 14. Anyway, to have a healthy functioning immune system, the main roles of the immune system is they're your surveillance system. And you have cells that will survey the body and they search for foreign invaders. Then they need to be able to recognize the foreign invaders. So there's the recognition step, and then they need to be able to destroy those invaders. So you can kind of describe the whole process of immunology as search, recognize, and destroy. But of course, there's very specific ways the immune system does that we will learn about. So first, searching and recognizing. How does the immune system know, moving my face around here, what is foreign? So the immune cells are your white blood cells, and there's a whole bunch of different white blood cells that we'll talk about, but I'll often abbreviate them as WBCs, so just keep that in mind. And all of these different white blood cells will patrol the body through the blood. There's some that reside in your tissues, and their job is to basically just be on the lookout for foreign particles. But they have to be able to recognize if something is foreign or if it's part of your body. And so body cells are known as self tissues, and foreign things are called non-self. Now, foreign things can be microbes, but they can also be foreign human tissues. Like if somebody gets a transplant, your immune system will often recognize those transplanted cells as foreign, which is kind of a negative because transplants are supposed to be helpful, but the body's job is to recognize non-self, and it does that very well. So the way that cells recognize each other, recognize things, is through cell surface markers, basically proteins and carbohydrates that stick off the surface of cells and basically act as name tags, like, hey, I'm Sarah's liver cell, or I'm Joanna's red blood cell, or I'm an E. coli cell. So all of these they're different chemical markers. They're like chemical name tags. And so when we talk about the word markers in cell biology, in microbiology, we're talking about things that stick off the surface of cells that kind of state their identity, that are identifiers, identifying molecules. That's what a marker is. So in this little picture here, we've got a white blood cell with some receptors, and these self cells that have these markers sticking off of them, and the white blood cell recognize them as self. All right, so if there's self cells, they don't have a reaction. But if the white blood cell recognizes them as foreign, a different reaction will happen. So these microbes, pathogens, tend to have some very general, easily recognizable features that we call pathogen-associated molecular patterns, or PAMPs for an abbreviation. So these are things that tend to be, for example, only on microbes, like only on bacteria. They're bacteria specific or viral specific. So for example, double-stranded RNA does not exist normally in humans. It's not a form that we store our genome in. That's something specific to double-stranded RNA viruses. So that might be something that that our white blood cells automatically recognize as foreign. Some other things, CpG is a chemical group that's on bacteria. Now I can't remember. I think CpG is a form of chemically modified DNA. There's also surface molecules on bacteria that fall into this category of PAMPs. I'm blanking right now on what all the different PAMPs are, but they are these just general patterns, like LPS, there's a good one, okay? Lipopolysaccharide does not exist in humans. It is endotoxin, found only in bacteria, gram-negative bacteria when they die, and so that is an automatic stimulator of our innate immune system. So innate immune cells have these receptors called PRRs, pattern recognition receptors, and they recognize these PAMPs, these different general patterns that are common in a lot of different pathogens. So you can think of our first line of defense, the innate immune system. They're kind of like the bouncers, I guess, of the immune system. They're just on the lookout. They're at the doorway. They allow, decide whether something is foreign, whether it comes in or not. So an important part of this first line of defense, or innate immune system, are these macrophages and monocytes. So it's a little confusing. Monocytes are what we call these cells when they're circulating in the blood, and then when they exit into the tissues, they become macrophages or other things. But basically, we have this system of macrophages, of phagocytes, let's put it that way, phagocytes, white blood cells that eat things, that live and permanently reside in all of our tissues. And they have different names in different tissues, but they essentially all arise from the same progenitor cell, and they all have the same function. So in the brain, they change, and these monocytes become something called microglia. In the liver, they're called Kupffer cells. In the spleen, we just call them macrophages. In a lot of tissues, we just call them macrophages. All right, but so there's this system within our body. If you look, like there's, kind of like in the lymphatic system, you've got lymph nodes everywhere. You also have pockets in all of your tissues where you store lots of these phagocytic white blood cells. So this whole system of tissues that all contain these macrophages or phagocytes is called the mononuclear phagocyte system, or the MPS. And again, this is just a system of phagocytes that are enmeshed in all the different tissues of your body, and their job is to be the lookout, to be the bouncers, to be the first to recognize foreign invaders. They're kind of out there with their binoculars looking around for PAMPs, for pathogen-associated molecular patterns. So the lymphatic system is really the system in the body that houses the cells of the immune system, all of our white blood cells. So we'll see that our white blood cells and red blood cells actually are all generated from the bone marrow. But the white blood cells, once they mature, primarily reside in the lymphatics. So it's a network of vessels that goes throughout the body, and its job is two parts. So of course it's important for the immune system, but another thing that it does is it removes excess fluid. So like water that's stored in the tissues can be removed via the lymphatics. We saw in a previous chapter somebody with elephantiasis, swollen legs because of a backup and buildup of fluid because the lymphatic vessels were blocked by a filarial worm. So the lymphatics are sort of a one-way flow. The circulatory system of the blood is a circular flow. You go from the heart to the lungs, to the heart to the body, to the heart to the lungs, to the heart to the body, and it's this constant cyclic movement. All right, but the lymphatics don't. They go from the tissues and they drain all that fluid into the blood, and then any excess fluid gets excreted through the urinary system. So it's sort of constantly draining in one direction. These dots, I don't know if you can see them that well in this picture, but some of these little grapes clusters that you see in the armpits, in the groin, you see some here in the abdomen, there's some in the neck. These are lymph nodes, and lymph nodes are basically just dense pockets of white blood cells. I kind of like to think of the lymphatic vessels as like train tracks, and the lymph nodes as train stations, where B cells and T cells can kind of hang out or get off and switch onto a different lymphatic vessel. So this is also why you find white blood cells in the blood, why they're called white blood cells, because they do circulate through the blood as well. There's far more in the lymphatic system, but there are some patrolling around in the blood as well. And when you have an infection, that number of white blood cells in the blood will go up. This is just to say that this picture is wrong. There is lymphatic vessels in the head and the brain as well. All right, so the parts of the lymphatic system include the fluid that flows through the lymphatic vessels, which is called lymph. The vessels themselves are very similar to blood vessels in their structure, and they branch into smaller and smaller vessels, ultimately lymphatic capillaries. The lymph nodes being oftentimes in clusters at joints, like in the armpits, the neck, in the groin, and they filter all that lymph fluid. Any bacteria or something that are drained from a tissue will go to the lymph node, and that's where there's a lot of B and T cells living. Some organs of the lymphatic system, the two organs of the lymphatic system, are the thymus, which is this fatty-looking organ that sits right on top of the heart, and it's really important because it's where T cells mature. And then the spleen, which is almost like a giant lymph node. It also happens to store extra blood. So like if you are bleeding internally, the spleen can sort of give you a little bit of a blood transfusion if you're hemorrhaging externally too, which is why if you damage your spleen, you get a lot of internal hemorrhaging because there's a lot of blood in the spleen, but also a lot of white blood cells. So in addition to the lymphoid organs and the actual lymphatic vessels and lymph nodes, we also have different pockets of tissues throughout the body where we have just dense clusters of immune cells, of white blood cells. And so they're considered part of the lymphatic system. So for example, your tonsils and adenoids, all right, in the throat and back of the nasopharynx, all right, remember the respiratory system constantly open to the air. You're constantly taking in food and air through your nose, through your mouth, and air through your nose. And that can contain lots of foreign particles, bacteria, fungi, viruses. So it makes sense that you have extra lymphatic tissue in that, back of your throat to help make sure there's a lot of white blood cells there to target any invaders. Okay. Another place is in the intestines. The GALT stands for the gut-associated lymphoid tissue. And so there's basically patches throughout the intestines that contain lots and lots of white blood cells. Because again, you have tons of bacteria in the intestines, many of which are supposed to be there, but potentially ones that are not supposed to be there. It can be a site for bacteria or other pathogens, viruses, to gain entry into the body. So it makes sense that we have extra defenses there. So that's what lymphoid tissue is. It's patches of tissue that are sort of embedded in other body systems that just give a sort of immune boost. All right, so we said that some of these white blood cells they're called white blood cells, so they do circulate in the blood. Most of your blood cells are red blood cells, or RBCs, but if we were to take the blood and we were to spin it down, get a sample of blood in a centrifuge tube, and then we centrifuged it, spin it, or just let it sit for several hours, it will separate into different layers. The top layer, that straw-colored fluid, is the blood plasma. This is basically just water with all kinds of nutrients and hormones and waste products and stuff dissolved in it. So it's the watery portion of the blood, and that's usually like 50 to 70% of the blood. Don't quote me on those numbers, but at least more than half of the blood is actually made up of the plasma. Your red blood cells here, the purpose of the red blood cells is mainly to transport oxygen. They're basically just packets of hemoglobin, and their purpose is to transport oxygen. So you do need a certain number of red blood cells in order to efficiently transport oxygen to your tissues. Move my face up here. So usually about a third to a half of the blood should be red blood cells. And then this tiny little band in the center here represents the white blood cells and platelets, which are like clotting factors. All right, so the white blood cells are really a very small portion of the blood, but that portion is larger if you have an infection or like a white blood cell cancer. So all of these blood cells, white and red alike, come from stem cells in the bone marrow. We call them hematopoietic stem cells, and they reside in the bone marrow, and they divide and differentiate and ultimately can become all these different types of cells in the blue boxes. So you don't need to know these different pink cells, the different stages of how they develop, but we are going to talk about pretty much all of these cells in the blue boxes. So red blood cells and platelets are found in the blood. Red blood cells are for carrying oxygen, and the platelets are involved in blood clotting. But all of these are the different types of white blood cells. So we'll talk about the different granulocytes, which all end in "phil," neutrophil, basophil, eosinophil, which has to do with actually the dyes they like to bind to. And we'll talk about the agranulocytes, the monocytes, which that's what they're called in the blood, but then they go into the tissues and they differentiate into macrophages and or dendritic cells. And these guys are all phagocytes. They can eat things. And then there's the lymphocytes, the B cells and T cells. And these are the ones that are involved in adaptive immunity. So I'll save them for the next chapter. But in this chapter, we're going to talk a lot about these guys, the neutrophils and the monocytes. So the second line of defense is nonspecific. It's innate because we're born with it. It's not something that you build based on your exposure to pathogens. It's something that you're born with. Your white blood cells already know how to recognize PAMPs. So the four key components of the second line of defense of innate immunity is phagocytosis, inflammation, fever, and antimicrobial proteins. And that's what we're going to focus on now. So phagocytosis, very important. It literally is a word that means a condition of eating cells. And that's what some of our white blood cells do, particularly neutrophils, monocytes, and macrophages. Again, I'll keep saying this, but monocytes and macrophages are kind of the same thing. They're called monocytes when they're in the blood, but then when they leave the blood to enter the tissues, they differentiate into larger cells called macrophages. But they all are types of phagocytes. They eat things. So this is actually a picture of a phagocytic white blood cell eating a yeast cell. You can see its pseudopods starting to wrap around that yeast cell, and it will ultimately engulf it. And then it will fuse with a lysosome and digest that cell that it's eating. So here's my image kind of explaining the monocyte and macrophage thing. So monocytes are what we call them when they're circulating in the blood, but when they leave the blood and go into the tissue, they differentiate, they grow into a larger cell, and then we call them macrophages, or sometimes we have other names for them like microglia or Kupffer cells, like we saw back on that page when we talked about the MPS, the mononuclear phagocyte system. Those macrophages in the different parts of the body do have different names. But for the most part, they're sort of the same cell at just different developmental stages or in different locations. So monocytes and macrophages patrol the blood a little bit, and they also patrol and reside in the tissues, and they're sort of like, I think, kind of like the bouncers. They're already there, they're present, they're already looking for invaders. All right. And so they're one of the first responders. Another first responder cell are the neutrophils. And neutrophils look like this. They're also sometimes called polymorphonuclear cells because they have, this is their, in purple here, this is their nucleus. You can see it's in many lobes. It's one thing, it's one compartment, but it's in a funny shape. So it almost looks like they have multiple nuclei. So they're very easy to identify on a blood smear because they have these multi-lobe nuclei. Anyway, these guys are the most abundant white blood cells in your blood. They are constantly patrolling. They're sort of like the EMTs, I think, of the immune system. And if there is a cut or a break to a physical barrier, neutrophils are the first on the scene. They will just run there and just start eating any foreign particles in that area. So they're already very common, but if you are having an immune reaction, if you have an infection, these numbers do go up. So how does phagocytosis work exactly? Well, the first thing is, so the white blood cells have to get to that site of damage or infection, and they do that through chemotaxis. So damaged tissue, we're going to see, releases chemicals that attract phagocytes, and they will move in a positive direction towards those chemicals, and we call that chemotaxis. Then they will need to stick to the wall of the tissue and enter the tissue, and also recognize that the foreign cell, and then they can phagocytosis, engulf it. And then they will fuse that food vacuole with a lysosome to digest the microbe, and finally the microbe is digested and eliminated, and they'll release the debris particles into the environment. Or, as we'll see in the next chapter, sometimes they will display them on their cell surface to show them to other immune cells. So this is just a visual of those different steps, which is probably better to look at than a bunch of words. So first thing it has to do is get to the site, chemotaxis, and then once it's there, it needs to recognize the microbe using the PRR. So this is the pattern recognition receptor on the white blood cell, the PRR. And these little things sticking off the bacteria are the PAMPs, the pathogen-associated molecular patterns. They get recognized, phagocytosis occurs, you get engulfment, and then the bacteria are inside this phagosome, like a vacuole, which then fuses with a lysosome. You can see that coming in, and those enzymes digest the bacteria, and then the waste is excreted, just released out of the cell. I'm going to show you this video because I think that I have this tendency to anthropomorphize and talk about bacteria like, you know, like they're little cartoony creatures, but this is really, it really does look cartoony in real life. So this is a video of an actual macrophage actually chasing, or I guess it's a neutrophil, chasing a bacteria.
In the midst of red blood cells. Staphylococcus aureus bacteria have been added. The small clump of bacteria release a chemoattractant that is sensed by the neutrophil. The neutrophil becomes polarized and starts chasing the bacteria. The bacteria, bounced around by thermal energy, move in a random path, seeming to avoid their predator. Eventually, the neutrophil catches up with the bacteria and engulfs them by phagocytosis.
I like the burp at the end. I always wait for it. All right, so they really do. They literally chase them down, recognize them, follow them via chemotaxis, chase them, recognize them, engulf them, and digest them. Alrighty, so again, that recognition occurs when the PRRs, the pattern recognition receptors on the white blood cells, recognize the PAMPs, those molecular patterns that are sticking off of the bacteria or virus particle. All right, so some bacterial PAMPs, some examples that I was trying to come up with before but was struggling. Peptidoglycan is a unique molecule that's found on bacterial cell walls. That can be a pattern that our cells recognize. Lipopolysaccharide, aka endotoxin. All right, and I was talking about double-stranded RNA is a classic viral PAMP. That's not something, double-stranded RNA is not normally found in eukaryotic cells. So phagocytosis was part one of the first line of defense of innate immunity. The second really important feature of innate immunity is inflammation. And inflammation is stimulated by a lot of those same cells. The macrophages and neutrophils can stimulate it, but also damaged tissue itself, damaged cells, body cells, skin cells, whatever, can elicit it as well. So the classical signs of inflammation, there's 5 of them. The first 4 that were identified in Latin in our from ancient Roman times, calor, rubor, tumor, and dolor, which stands for heat, redness, swelling, and pain. And then the 5th one that's sometimes used as well is loss of function. So generally, if you have pain and swelling, you also can't use, if your arm is painful and swollen, you also can't use it. But so these first 4 are really the most important ones, the things we see: heat, redness, swelling, and pain. And those things are in response to trauma. And those signs of inflammation, the heat, redness, swelling, and pain, can be local. Like if you get a cut, you can get localized swelling and heat and pain and redness. Or they can be systemic, like if you have an infection internally, or if you have septicemia, in a blood infection, then you can start getting basically, your whole body starts experiencing pain and swelling and redness and heat, okay? So inflammation, chronic inflammation in localized areas can be very damaging. And this is really the heart of heart disease, pardon the pun. A lot of people think it comes from diet and stress, and those things can contribute to the development of cardiovascular disease. but what researchers have realized is really the starting point of cardiovascular disease is inflammation of the blood vessels. When they become inflamed, then you can start building up plaques on those particular spots of the blood vessels. Ulcers can be due to chronic inflammation. Cancer can be due to chronic inflammation. So there's it's a adaptive response in that we use to try to kill bacteria and viruses and foreign particles. But if it's on all the time, or it's on chronically, it can also lead to organic diseases. So inflammation is a good thing, but too much inflammation is a bad thing. So the purpose of inflammation in the immune system is to attract white blood cells to the scene to come and help fight the pathogen, and to help set in motion mechanisms for tissue repair from the injury of all the inflammation, and of course to destroy the microbes themselves and prevent them from further invasion. So these chemicals that are produced that cause all of the redness and swelling and pain, etc., are called cytokines. Cytokines are just chemicals that function in the immune system. So hormones are also chemicals. They're chemical messengers that our glands secrete to signal things. And we have neurotransmitters, which are the chemicals that are secreted by our nervous system, our nerve cells. So it's sort of a common theme in anatomy and physiology that cells secrete chemicals to do things, to basically send messages. And in different systems, we give those different chemicals different names. So cytokines, hormones, neurotransmitters, they're all very similar molecules. They have different functions. So cytokines are the names of the chemicals that have functions in the immune system, that signal immune cells, that signal inflammation. So there's a bunch of different cytokines, just like there's different hormones, different neurotransmitters. We won't get into all the different cytokines, but in general, if I'm talking about cytokines, I'm talking about chemicals that are secreted by immune cells to communicate information about infection and inflammation. So these are some of the many cytokines that are produced. Some that I want to call your attention to that we will talk more about. Interferons. So interferons interfere with the ability of viruses to infect a cell. So there's different classes of interferons. But the ones that we will focus on are ones that are produced by damaged cells that have been infected by a virus. They can basically produce these interferons to tell other cells, hey, there's viruses around, like shut down your borders and don't let the viruses in. Some other ones that we'll talk about that are really involved in inflammation, histamine, serotonin, and bradykinin are all involved in the redness, a lot of the redness and swelling reactions that are part of inflammation. Prostaglandins are ones that produce pain, and pyrogens, which are not on here, are ones that produce fever. So these are just a bunch of popular cytokines. You won't need to know all of them, but we will talk about some of them further. All right, so the stages of inflammation, you get an injury. All right, let's say you get a splinter or a cut in your skin, and that thing that you cut yourself with contains some bacteria on it. So those bacteria have now made it past that first barrier of your skin, and now it's time for the second line of defense, the innate immune system, to take care of things. So when you damage these cells through that injury, those cells will start to release these cytokines, basically like SOS signals to the innate immune system, like, help, we've been damaged. And so those signals go to the blood, and then white blood cells flowing through the blood, primarily those neutrophils, they sort of smell those chemicals, and they exit the blood vessels into the tissue so they can start doing phagocytosis. As they're exiting the blood, they also fluid leaks out with them. And so that's where you get the swelling from, the fluid entering that tissue along with those white blood cells. You may develop pus at the site, and pus is just basically a bunch of dead neutrophils. And then once those neutrophils are done, because they eat bacteria and then they die. But they also will secrete chemicals that help to stimulate healing of that tissue. And then that's inflammation. The inflammation process is done. So you have a series during inflammation of blood vessel dilation and blood vessel contraction. That dilation stage happens early on because these white blood cells, they need to leave the blood vessels in order to go into the tissue. They also need to exude that fluid to cause the swelling. All right, but initially, I just lost my place. Where was I? Well, now I can't remember when it closes off. But anyway, lost myself there. When the white blood cells are leaving the blood vessels, they sort of squeeze through. Like, I don't know if you've ever seen those videos of like an octopus getting through like a tiny little opening, but that's what it looks like when white blood cells squeeze through these tiny openings. And that process of squeezing through those tiny openings is called diapedesis. That's just a vocabulary word. So diapedesis is the squeezing through of white blood cells through capillary pores to get into the tissues. All right. This is just a very complex and detailed flow chart of how inflammation works. So you get tissue injury, all right? And one of the things that does is it releases cytokines in that damaged tissue that will cause the blood vessels to dilate, which allows fluid to leak into the tissue. It also causes that swelling and pain from the pressure. All right. It also can release cytokines that lead to the heat and redness of that tissue site. And it also leads to attraction of neutrophils, white blood cells, to the site where they migrate to the injured area, and they can do diapedesis, exit the blood vessels, and then do phagocytosis, eating the bacteria in that area. And all of this together leads to healing of that wound and prevention of infection. So the inflammatory response, can't get my face quite out of the way here. The stages are, first, tissue injury. So you get a cut or a bacteria invades and does injury with enzymes. All right, so you're going to have cytokines that are released by those cells, those injured cells, to attract neutrophils, white blood cells from the blood. So the vascular reactions, what happens is you get that vasodilation, so the blood vessels widen to allow those white blood cells to move out to do diapedesis. And my video here kind of shows that. Oh, never mind, it's not available. Well, might as well have just thrown in a video of an octopus squeezing through a small hole, and I recommend that you Google that because it's pretty entertaining to watch. This is just another slide showing you diapedesis from a cartoon version and also from an actual electron micrograph of blood cells that are leaving the blood vessel doing diapedesis. All right, so in addition to the white blood cells leaking out of the blood vessels, you also get fluid that's leaking out, and swelling of a tissue. Excess fluid in a tissue is called edema. It's the medical term for it. And the edema, it helps to sort of build up pressure in that site, so it kind of traps the microbes so they can't move. And it that's pretty much the point of it. If it's pussy, if there's like that whitish greenish color to it, that's actually dead neutrophils that are building up. Fever is another classic sign, heat. Fever, heat is another classic sign of inflammation. Fever is a systemic response that's controlled by the hypothalamus in the brain, which regulates body temperature, and it's stimulated by chemokines or cytokines called pyrogens. Pyro means fire, so pyrogen is a chemical that initiates fire or fever. So fever is often a lot of people are afraid of fever. If you have a high fever, like they rush their kid to the doctor's office. And there are some negative effects of fever itself. So children, in particular those under 5, can experience something called febrile seizures, where an elevated temperature causes them to have seizures. These seizures, however, are not they're not damaging to the brain like epileptic seizures can be. So they're not dangerous. They're scary as hell, but they don't cause any long-lasting damage. So fever, the thing about fever is it's actually adaptive. The purpose of fever is it actually increases your immune cell replication, so your immune cells replicate faster at higher temperatures, but pathogens replicate slower at those higher temperatures. So it is an attempt for your body to mount an effective immune response. So if you can handle it, sometimes they say it's better to not treat a fever, but if it's getting very high, especially in a child, you definitely want to control that fever with antipyretics, some, Advil, Motrin, Tylenol, whatever the antipyretic of choice is. But fevers in and of themselves are not usually considered dangerous. They have to be extremely high in order to be, I think, over 105 is starting to get concerning, but a fever of 101, 102 is not usually something to be concerned about, and actually might be beneficial. Just usually, along with fever, you feel pretty crappy. So most people will treat their fevers to make those crappy feelings and symptoms go away. But the fever is often a sign of an infection because it's a result of pyrogens being produced by white blood cells in response to a foreign cell, a foreign pathogen. So you can treat fevers with antipyretics, which are over-the-counter medications that are fever reducers, and other things that can go along with fevers besides the aches and pains. It can also increase your heart rate and your breathing rate. When my daughter, one time when she was young and she had an infection and she had a high fever, and I started to get kind of nervous because I noticed her breathing was very fast. But that's a normal sign of fever, and once the fever was treated, she went back down to normal breathing. One of these cytokines that I was telling you about that I will tell you about are these interferons. So interferons, there's different classes. You've got interferon alpha, interferon beta, and interferon gamma. So alpha and beta are the ones that we're going to talk about in this chapter. And the reason it's called interferon is because it interferes with virus's ability to replicate. So you have a cell on the left here. This one is it's been infected with a virus. All right, and it's assembling viral proteins. It has been hijacked by this virus. It is already a slave to the virus. All right, but at the same time, it is sending out a warning signal. These green balls represent interferons, and those are being received by a neighboring cell that hasn't been infected yet. And it's basically telling this cell, do what you can to block yourself from being infected. There's a virus around. And so it can start synthesizing antiviral proteins or stop synthesizing certain receptors. Basically, it gives the other cells in the surroundings warnings so they can mount some defenses. All right. Another element of, the last element of the innate immune system is antimicrobial proteins. So far, just a reminder, we have phagocytosis, inflammation, fever, and antimicrobial proteins are the 4 elements of the innate immune system. So these antimicrobial proteins are just small proteins that actually help to recognize, lyse, destroy bacteria. And one important family of these antimicrobial proteins is called complement. And I think it's the stupidest name for proteins. It always confused me in my immunology classes. I'm like, complement, like, it's like you give someone a compliment. That's like a nice thing to say, even if that's not the right way to spell it. But it just seemed like a weird name. So the reason it's called complement is it's a family of proteins and they complement the immune system. Like, they complete it. They're like an awesome sidekick. I don't know, still a stupid name. It's a family of proteins. There's like 30 proteins, complement 1, complement 2, complement 3b, etc., etc. And they can do a bunch of things. The two most important things that they do is, one, they can do opsonization. And all right, so opsonize is a word that I like to say it means to candy coat the bacteria. So to opsonize, it just basically means to bind all over the surface of the bacteria and make the bacteria look especially delicious for phagocytes and attract phagocytes to the bacteria. So I like to think of opsonization. Another protein that can opsonize or coat bacteria and make them look really yummy for phagocytes are antibodies, which are part of our adaptive immune system. But complement is a type of candy coating that's part of the innate immune system. These complement proteins just circulate in the blood, and they sort of recognize PAMPs, very general features of bacterial cells, and can bind to them. And then phagocytes can see them more clearly and eat them. Right? Another really important thing that complement can do is they can form these, like, literally they can assemble these hole punchers that punch holes in the walls of bacteria, and these are called MACs, or membrane attack complexes. And I still remember my immunology professor calling it a Mac attack, which sounds like a hankering for a hamburger from McDonald's, but it's actually poking holes in bacteria. So you get a bunch of these complement proteins that can come together and literally form this hole puncher. You can see it in this electron micrograph here, and it just stabs bacterial cells and then pops them, essentially. It makes giant holes in their membranes, and fluid starts leaking out of the cells and they die. Or fluid starts entering the cells and they burst, either way. So I call that a MAC attack, the membrane attack complex. So the two ways that complement really helps to complement the immune system is to opsonize, or candy coat, bacteria and make them really delicious for phagocytosis, or MAC attack, form these hole punches and punch holes in bacteria. In addition to the complement proteins, there's other antimicrobial proteins that we produce in our skin, in our blood, that we're just starting to understand. And some of them are potential drug categories, new antibiotic classes that are being examined as new antibiotics. But a lot of these antimicrobial proteins they're not even proteins, they're really short peptides, meaning they're made of only 15 or 20 amino acids, and they can disrupt bacterial membranes. They have a lot of different names: defensins, protegrins. You don't need to know all of those, but I'm just mentioning it because they do exist and they are an area of research. But complement is definitely the main family of antimicrobial proteins. And that is the end of this chapter. Sorry it's so long. Of course, you can watch it on a slower speed or pause, rewatch as many times as you need. Bye.